Welcome back to Clinical Hematology Reviews. Today, we'll be discussing lymphocytes. In healthy adults, lymphocytes constitute the second most abundant leukocyte population in peripheral blood, accounting for roughly 20 to 40% of total WBCs. Morphologically, circulating lymphocytes appear relatively uniform under routine right seamless staining, but functionally, they are divided into three distinct lineages. These subsets include B lymphocytes, T lymphocytes, and natural killer or NK cells.
That functional diversity underpins our adaptive immune defense and innate surveillance. The canonical maturational sequence progresses from the lymphoblast to the prolymphocyte and ultimately to the mature lymphocyte. However, in routine clinical practice, distinguishing a lymphoblast from a prolymphocyte based strictly on light microscopy can be exceptionally difficult. Because of this morphologic overlap, clinical laboratories routinely categorize precursor lymphoid cells broadly as blasts or mature lymphocytes, relying on immunophenotyping for definitive staging.
That is a crucial operational distinction for laboratory workflow. While morphologic evaluation provides immediate clues, immunophenotypic markers like CD34 and CD45 are essential for definitive identification. Functionally, lymphocytes serve as the primary cellular drivers of adaptive immunity, providing antigen-specific recognition, targeted effector responses, and lifelong immunological memory against foreign pathogens.
Let's examine the detailed morphologic criteria across the maturation stages, beginning with the lymphoblast. The lymphoblast ranges from 10 to 18 micrometers in diameter and exhibits a very high nucleus-to-cytoplasm ratio. Its nuclear chromatin is loose, fine, smudged, or described as lacy, usually containing one or two prominent nucleoli. The cytoplasm is scant, agranular, and light blue, lacking the primary granules seen in myeloid precursors.
Cytochemically, lymphoblasts stain positive for PAS, or periodic acid Schiff, and TDT, which is terminal deoxynucleotidyl transferase. Crucially, lymphoblasts are strictly negative for myeloperoxidase, or MPO. This negative MPO reaction is a key laboratory rule used to differentiate lymphoid blasts from myeloid blasts in acute leukemias. Immunophenotypically, blasts express the primitive stem cell marker CD34 alongside the pan-leukocyte marker CD45.
As the cell transitions to the prolymphocyte stage, it becomes slightly smaller than the lymphoblast while retaining a high nucleus-to-cytoplasm ratio. The nuclear chromatin becomes moderately more condensed than that of a blast, typically displaying a single prominent nucleolus. The cytoplasm remains agranular and light blue, lacking the primary granules seen in myeloid precursors.
Finally, the mature resting lymphocyte is quite small, roughly the size of a surrounding normal red blood cell, measuring around 7 to 10 micrometers. Its nucleus occupies approximately 90% of the overall cell area, demonstrating a high nucleus-to-cytoplasm ratio. The chromatin is dense, dark purple, and heavily clumped, with nucleoli being extremely rare or invisible under light microscopy.
The cytoplasm of a mature resting lymphocyte forms a narrow sky blue rim around the condensed nucleus. While typically agranular, occasional nonspecific azurophilic granules can be identified in a small percentage of resting cells. Loss of CD34 expression confirms complete maturation, while CD45 expression is strongly maintained across all mature peripheral blood leukocytes.
Moving beyond standard resting morphology, we occasionally encounter large granular lymphocytes, or LGLs, in peripheral blood films. Morphologically, LGLs are larger than resting small lymphocytes and feature a round or slightly indented nucleus with condensed chromatin. Their cytoplasm is pale blue and more expansive, characteristically packed with prominent coarse azurophilic granules.
From a lineage perspective, LGLs represent a specialized circulating subset derived strictly from T-cell or NK cell populations. B lymphocytes do not assume this granular morphology under normal or reactive conditions.
When assessing LGLs, immunophenotyping typically reveals markers such as CD3 for T-cell variants or CD16 and CD56 for NK cell variants.
Next, let's analyze reactive lymphocytes, which are also known as atypical or variant lymphocytes. These cells represent antigen-stimulated immunocompetent B or T cells, though the majority circulating during acute viral infections are activated T cells. Clinically, they are classically observed in high numbers during Epstein-Barr virus infections, such as infectious mononucleosis.
Morphologically, reactive lymphocytes are significantly enlarged, ranging from 16 to 30 microns in diameter, and exhibit a decreased nucleus-to-cytoplasm ratio compared to resting lymphocytes. The nucleus is highly variable in shape, presenting round, lobulated, folded, or elongated contours with chromatin that ranges from fine to moderately clumped. Nucleoli may occasionally be visible due to active protein synthesis.
The cytoplasm of a reactive lymphocyte is abundant, amoeboid, and displays variable basophilia. A classic diagnostic hallmark is localized peripheral basophilia, where the cytoplasm stains intensely dark blue along its outer edges. Furthermore, the flexible cytoplasmic membrane characteristically molds around or hugs adjacent red blood cells, creating a dark, scalloped border.
That classic hugging feature helps differentiate reactive lymphocytes from both blasts and monocytes on a peripheral smear. Blasts possess smooth, rigid cell borders and higher nucleus-to-cytoplasm ratios, whereas monocytes feature dull gray-blue ground glass cytoplasm with fine dust-like granules and vacuolation. Recognizing these subtle cytoplasmic contours prevents the misclassification of a benign viral response as acute leukemia.
When B lymphocytes undergo terminal differentiation following antigen stimulation, they transform into plasmacytoid lymphocytes and fully mature plasma cells. These specialized cells are dedicated exclusively to mass antibody synthesis and secretion. On a peripheral blood smear, mature plasma cells measure 16 to 20 microns in diameter and feature a distinctly eccentric nucleus.
The nuclear chromatin of a plasma cell is dense and heavily clumped into a characteristic cartwheel or clock face arrangement. The cytoplasm displays deep, intense basophilia due to an abundance of rough endoplasmic reticulum required for protein production. Right next to the eccentric nucleus, a prominent clear area known as a perinuclear halo is visible, representing a highly developed Golgi apparatus.
Pathological or hypersecretory conditions can induce striking morphologic variations in plasma cells, such as flame cells and Mott cells. A flame cell exhibits vibrant reddish-purple or pink staining cytoplasm caused by an extraordinary intracellular accumulation of immunoglobulins, most commonly associated with IgA heavy chains.
Conversely, a Mott cell, sometimes called a grape cell, features cytoplasm completely packed with clear spherical inclusions known as Russell bodies. These Russell bodies represent localized globules of synthesized condensed immunoglobulins that have accumulated within the rough endoplasmic reticulum. Identifying these plasma cell variations provides immediate morphologic evidence of monoclonal or polyclonal hypergammaglobulinemia.
Now let's examine lymphocyte kinetics, distribution, and reference intervals. Age is a major variable in clinical interpretation, as young children present with significantly higher relative and absolute lymphocyte counts than healthy adults. In infants and early childhood, the relative ratio of neutrophils to lymphocytes is naturally inverted compared to adult baseline parameters.
Anatomically, only about 5% of the body's total lymphocyte pool circulates within peripheral blood at any given time. The remaining 95% is sequestered in extravascular lymphoid tissues, including lymph nodes, the spleen, mucosa-associated lymphoid tissue, and lymphatic fluids. Peripheral blood lymphocytes are further divided by lifespan into two distinct dynamic populations.
Approximately 80% of circulating lymphocytes are long-lived memory cells that continuously recirculate between blood and tissues for months, years, or even decades. The remaining 20% consist of short-lived effector cells that persist for only a few hours, up to 5 days. This kinetic balance ensures immediate effector readiness alongside durable immunological memory.
In terms of absolute quantitative thresholds, lymphocytosis in adults is defined as an absolute lymphocyte count exceeding 4.8 times 10 to the third per microliter. Conversely, lymphopenia is defined as an absolute lymphocyte count falling below 1.0 times 10 to the third per microliter. Both conditions warrant targeted clinical investigation to identify underlying infectious, autoimmune, or malignant causes.
Functionally, lymphocytes represent the primary effector cells driving immune responses against viral pathogens, intracellular bacteria, and transformed neoplastic cells. They execute three interconnected branches of immunity: humoral immunity, cell-mediated immunity, and innate immune surveillance. Each functional subset relies on specific surface receptors and cluster of differentiation markers.
Humoral immunity is mediated by B lymphocytes, which differentiate into plasma cells secreting antibodies upon antigen engagement. Cell-mediated immunity is directed by T lymphocyte subsets, including CD4-positive T helper cells, CD8-positive T cytotoxic cells, and regulatory T cells. Finally, innate cell-mediated killing is executed by natural killer cells, which rapidly eliminate compromised target cells without prior sensitization.
Focusing on B lymphocytes, they constitute 15 to 30% of the circulating peripheral blood lymphocyte pool. Immunophenotypically, B cells are defined by the co-expression of surface markers CD10, CD19, and CD20. Their central physiological role is the production of immunoglobulins or antibodies, which neutralize soluble antigens and opsonize pathogens.
Structurally, every monomeric immunoglobulin molecule consists of two identical heavy chains and two identical light chains. The heavy chains determine the antibody class, designated as IgA, IgD, IgE, IgG, or IgM, while the light chains are classified as either kappa or lambda. Naive B cells express surface IgM and synthesize large pentameric IgM molecules during the primary immune response.
Upon secondary antigen exposure, activated B cells undergo immunoglobulin class switching to produce monomeric IgG, which mediates long-term systemic adaptive immunity. Memory B cells maintain this class-switched surface immunoglobulin for rapid recall upon reinfection, while terminally differentiated plasma cells secrete massive quantities of soluble antibodies directly into circulation.
Turning to T lymphocytes and natural killer cells, all mature T cells are identified by the pan-T cell marker CD3. T lymphocytes interact directly with antigen-presenting cells, or APCs, such as macrophages, and secrete cytokines to orchestrate immune activation. T helper cells express CD3 and CD4 while lacking CD8, recognizing processed peptides presented on MHC class II molecules to direct responses against extracellular pathogens.
Conversely, T cytotoxic cells express CD3 and CD8 while lacking CD4, recognizing peptides presented on MHC class I molecules to eliminate intracellular pathogens and virus-infected host cells. Natural killer cells lack CD3 but express CD16 and CD56. As key effectors of innate immunity, NK cells recognize and lyse virus-infected or malignant cells without MHC restriction, releasing interferon gamma to activate surrounding macrophages.
In summary, we have detailed the complete maturational sequence, distinctive morphological variations, kinetic parameters, and immunophenotypic profiles of circulating B cells, T cells, and natural killer cells.
Thank you for joining us, and we look forward to bringing you our next high-yield review.